Hello everybody, thanks a lot for attending my virtual talk. Today I'm going to speak about the semi-synthesis of novel probes to study bacterial glycolipid processing enzymes, and I'm Dr. Stephen Cochrane from Queen's University Belfast. Okay, so our research in the Cochrane lab is geared towards the challenge of antimicrobial resistance. Of course, we've all heard about the problems posed. If resistance is left unchecked by the year 2050, it could cause over 300 million premature deaths and cost the global economy on over 100 trillion US dollars. Therefore, much needs to be done to identify new antimicrobial compounds and antimicrobial targets. Bacteria produce a number of essential glycopolymers. On the left-hand side of this slide, we have a cross-section of a gram-negative bacterial cell. So for example, in gram-negative bacteria, on the surface of the outer membrane, you have lipopolysaccharide. This is a polysaccharide chain that protects cells from bile salts and lipophilic antibiotics. Both gram-positive and gram-negative bacteria have peptidoglycan. This is a glycopolymer that provides extra structural strength to the cell, as well as balancing osmotic pressure of the cytoplasm. Also in bacteria, you have N and O-linked protein glycosylation, which is an essential pathway and has a number of cellular functions. When you take a look more specifically at bacterial glycopolymer synthesis, you start to see some common themes. In the middle here with the red arrows, we have peptidoglycan biosynthesis, and I should state this is a fictionalized membrane because it has enzymes from both gram-positive and gram-negative bacteria. So starting from undecaprenol phosphate, which is the universal lipid carrier in bacteria, a series of glycosylations yields lipid 2, and then ultimately this is flipped across the cell membrane before being elongated into peptidoglycan. In the synthesis of the O antigen subunit in lipopolysaccharide, this again starts from undecaprenol phosphate, and a series of carbohydrate extensions yields the desired monomer, which is then flipped and polymerized before being incorporated into lipopolysaccharide. And again, a similar series of reactions with transglycosylases takes place in the synthesis of teichoic acid in gram-positive bacteria. So all of these glycolipids shown on the bottom contain an undecaprenol chain, and in this talk are going to be called undecaprenol containing glycolipids. So these are essential biomolecules in the biosynthesis of bacterial glycopolymers, and they're present in many of the major pathways for making glycopolymers. So because undecaprenol is common in many important different glycopolymers, a lot of enzymes that are potential antibiotic targets process undecaprenol containing glycolipids, and therefore the undecaprenol chain would be a great site for modification, because you can imagine if you take one undecaprenol and you were able to stick a chemical label here, say for example a fluorophore or photoaffinity tag, now all of a sudden you have one undecaprenol analog which could be used to synthesize multiple different UCGs. So there's a bit of a catch here. Undecaprenol is incredibly expensive. It costs over €400 for 10 milligrams. So previous efforts making labeled undecaprenol have focused on either total chemical synthesis or biosynthetic assembly. So for example, Chuyi Wang's lab have looked at the total synthesis of polyprenols containing various labels. This works well. However, some of the limitations are it produces only multi-milligram quantities. It's also incredibly labor intensive. It uses air and moisture sensitive reactions. So really it's only possible for a skilled synthetic chemist to make these types of analogues. And again, this hexaprenol analog is only half the length of undecaprenol. Jerry Troutman and co-workers have used a biosynthetic assembly approach where they start from nerol. In 7 steps, they make a labeled farnesyl pyrophosphate analog and then use the enzyme undecaprenol pyrophosphate synthetase to produce the full-length labeled undecaprenol pyrophosphate. This is shorter However, because it uses enzymes, it has limitations in terms of scale and may be substrate limited. Also, the product you get, this needs to be hydrolyzed if you want to use this in glycopolymer synthesis to the phosphate derivative. So there was a third strategy that hadn't been considered in the literature, and this was direct labeling for undecaprenol. So the inspiration from this project came from one of my main hobbies, which is long distance running. And let me just tell you a bit about Rosie Ruiz. So she was the female winner of the 1980 Boston Marathon. And this is the Boston Marathon course shown here. So there's a number of ambiguities about Rosie winning the 1980 Boston Marathon. When she finished, she was neither panting or coated in sweat, both of which I am when I finish. She managed to improve her PB by 25 minutes from the New York City Marathon, which she also cheated in. She was caught taking the subway during this, and no spotters during the race actually remembered seeing her. So it turns out that 2 students reported seeing her jump out of the crowd onto the course at around 0.5 miles from the finish. So she really didn't run the marathon. She ran the very last bit. So she just snuck in right at the end. And that made us think, okay, could we take undecaprenol and do some type of late stage functionalization right at the end, ideally in a position far removed from where enzymatic processing takes place. So to do this, we needed a healthy supply of undecaprenol. Bacteria produce undecaprenol. It has the Z8E2 omega configuration. However, this is present in very low amounts and is not easily isolated. Thankfully, plants are nice. They produce lots of polyprenols, and in particular bay leaves produce a closely related analog to bacterial undecaprenol. So it differs just by the configuration of one of the internal alkenes. And almost all studies published use plant undecaprenol because this is what you buy from companies like Lorodon and American Radiolabeled Chemicals. So we developed a optimized large-scale extraction of undecaprenol. So we buy bay leaf powder, only 15 pounds for about 2 kilos. We add this to Socrates, our big soxylate extractor, and we sequentially extract 2 1 kilogram portions each for 48 hours. We then detach the soxylate extractor. We add some potassium carbonate and methanol in with the extract and stir that over 72 hours. This increases the yield of polyprenols by methylizing any esters, and it also is reported to remove phenolic impurities. At this stage, we then do a partial purification to get crude undecaprenol, about 16 grams in this case. And so the material you get isn't particularly high quality, but we found rather than doing additional purifications with silica or by reversed phase, if you take this partially purified material and then acetylate, you can then get some nice high quality material. And we were ending up with typically about 10 to 12 grams of this per extraction. So really a sizable quantity that we could then start making undecaprenol analogues with. So we then considered what positions we could modify on undecaprenol. The alpha isoprene unit, this is the hydroxyl, which could give a directing effect. So this would be the easiest. However, this is also the position where most of the business happens. So you form the phosphate in this end, and then glycosyl or phosphotransferases synthesize undecaprenol containing glycolipids here. So we imagined if you stuck a really bulky label at this position, it probably would be a bad idea. So we focused then just on small labels, and the first thing we tried was putting something like deuterium. And so this is quite straightforward to do. You can take manganese dioxide. It also works with other oxidants like PCC, and you can oxidize to the aldehyde, and then we do a Lusch reduction with sodium borobutyric to give this monodeutyrated undecaprenol analog. Using standard phosphoramidite chemistry, this can then be converted into deuterated undecaprenol phosphate. And before COVID hit, we were in the process of sticking a CF3 label into here. So in place of deuterium, because we're interested in looking at some enzymes that process UCGs by fluorine NMR, and this will be done hopefully when we get back in the middle of July. So we figured you can't really stick big labels in the alpha position, so let's try somewhere else. The obvious position would be the omega position, but we were kind of unsure how we could modify one tri-substituted alkene in the presence of all the others. So another, 9 are in here. But there was some literature precedent for this. And so we adapted the conditions, and gratifyingly we found that if you start with undecaprenol acetate, you can get formation of this epoxide over 3 steps in 42% yield. And then very quickly you can cleave this epoxide using periodic acid to get the aldehyde, which acts as a handle then for further modification. And so we tried a few different approaches. We initially tried Wittig chemistry, but this was quite cumbersome. And we finally focused on using reductive amination. And using this approach, we incorporated a variety of labels, so things like fluorophores, photoaffinity labels, bioorthogonal tags, so for further functionalization, and then things like spin labels, which could be useful for studying these transmembrane proteins by things like tryptophan quenching. We then wanted to test were these modifications having any effect. So the enzyme that works on undecaprenol is undecaprenol kinase. It's found in gram-positive bacteria and has some implications in resistance to things like antibiotics like bacitracin. So we used the standard pyruvate kinase lactate dehydrogenase coupled assay to look at enzymatic activity. And so whenever we have natural undecaprenol, we got a value of about 8.5. And as you can see from all the other various analogues, pretty much everything is reacting in very similar rates to the natural product. The exception is whenever we have an alcohol at this position, and this is protected as a THP ether. So we've effectively flipped undecaprenol, so the order of the E and Z double bonds are reversed. And this reacts in about half to a third of the rate. So we were quite happy with this. And then in the kind of final effort towards at least making something publishable, we decided we needed to show that we could put these analogues into glycolipids. So my lab works a lot on lipid 2 binding antimicrobial peptides. So we had some of this intermediate lying around in the lab that is the penultimate intermediate in lipid 2 synthesis. And so taking this precursor, the benzyl protecting groups in the phosphate are removed by hydrogenolysis. We can then couple the deuterated undecaprenol phosphate and then do a global deprotection. And after HPLC, we had about 22% yield over 4 steps, which corresponded to about, I think it was 15 milligrams of material, starting from 50 milligrams of this intermediate. And this, I think, highlights nicely the power of this method because you're sticking the polyprenyl chain in at the very end. So it's a very efficient method for chemical labeling. Okay, and so that is just a summary of our work. So we were able to successfully extract undecaprenol from bay leaves, and using chemical synthesis, we can make a series of nice labeled analogues, which we hope will be useful for people who are working with enzymes that process these various different substrates. Our work was just published online at the end of last week. So what was that, like the 1st of June, I think. So if you're interested in the work, you can see more in our paper. And so there's the references published in Chemical Communications. And with that, first of all, I'd just like to thank the conference organizers for putting together this virtual conference and for giving me the opportunity to speak. In this project, Francesca, who's not in the photo, she was the undergrad who started this project. Dr. Rachel Cochrane was the one who really led this work, and yeah, then I also have my other group members. This is us in our Christmas party photo. I'd like to thank Martin Caffrey. Martin in Trinity College Dublin were the lab who did the testing with undecaprenol kinase. And thank you all for listening, and during the actual question period I will be happy to answer any questions.